So I can check the time that I have. I will do the opening and then I call you. It's okay? I leave it here.
Só preciso, já pode pôr uma delas. Eu só preciso compartilhar.
Se eu conseguir colocar a tudo completo do simpósio, porque aí eu vou fazer a abertura, aí na sequência eu já chamo ela.
Tá, só a imagem do simpósio.
Porque 9 horas começa, vou falar, vou fazer a abertura.
Deixa eu...
No, we do it with one spec.
Okay, because I'm sending a quotation to see if I will send the samples to them, or yeah, I would like to work with the tryptophan metabolism and making that way.
We are now doing
Okay, I know that is. Okay. Okay.
So we did it, we didn't have to start with Canada. And now we are going to
remember that we published that English and aging paper? Yeah. If you want, we have all the data, and we have an excellent partner that is a doctor that works with this, and he's very nerd, and yeah, he's amazing. So we can look specifically to your, yeah, if you want, because we can compare. Because this paper came from, I did my alone work with him, and the comparing of controls, it was black and white. And one thing that I asked him is it due to tumor or the age is relevant here? So that's, we took all the controls that we had, like 170, and we separate by age. And for tryptophan metabolism, for example, after 80 years old, there is a shift. So that's why I'm interested in doing this. But he has everything. So we can look at the aspect that is numbers. Yeah.
because now we have to Good morning to all of you. I'm Valkyria Bueno from the Department of Microbiology, Immunology, and Parasitology, and it's a great pleasure to give you my welcome to the Symposium Aging and Future Challenges. So this symposium is funded by PrInt. So with PrInt project, we were able to bring here today 3 professors working on the aging field. So we have Professor Daniela Frasca from the University of Miami, Miller School of Medicine. We have Professor Gisele Colleoni from Medicine Department of Unifesp and Geriatrics and Gerontology Division. And we're going to have also Prof.Ligiana Corona from Unicamp. So as you know, people older than 60 years are growing in number in all over the world. And they can have several diseases, chronic diseases, that can impact their lives, the daily basis life. And also is an impact on the health system. For instance, in Brazil, we're not ready to have this high financial cost that old people can bring with these diseases. So this mini symposium is going to reunite people studying and working on aging field. So we're very thankful for all of you that are on the audience or on the YouTube following us. And after each speaker, we can have 10, 15 minutes for questions and discussion. And either you can ask the questions in English or in Portuguese. Feel free to talk with the speakers. They are very nice and they can answer all your questions, okay? So thank you. And now I'm gonna call the first speaker, which is Professor Daniela Frasca from the University of Miami. And she's gonna talk.
Preciso compartilhar no YouTube. Espera só um momento. Fechando. Essa aqui. Essa.
Okay. So it's the third time Professor Daniela Frasca is at Unifesp, thanks to PrInt, and now she's going to give us her talk. And so thank you, Daniela, for accepting this third invitation to be here at Unifesp, and you're very welcome to give your talk.
Thank you very much, Valkyria, for the invitation to be here and for making this third trip to Unifesp possible. Thank you for the people that are here today, either in presence or in the YouTube. And I would like to introduce myself, saying that all my career I have been working on the effects of aging on humoral immunity, the effects of aging and age-associated diseases on humoral immunity. And very recently, I became very interested on the immunometabolic regulation of humoral immunity. So first of all, my slide with acknowledgements. I'm very thankful to the talented students that are here in red. They did all the experiments that I'm going to present today. I'm a very basic science person, and I'm only doing basic science or preclinical studies. I have to thank the doctors that allowed the selection and recruitment of the people that have donated their blood to do the experiments that I'm going to show you today. And of course, Valkyria, now long-time collaborators, even before this possibility to come to Brazil, the facilities in our medical campus, and of course, the funding, especially the NIH with a recent R1 and the CAPES from Brazil. Again, I became very interested in the metabolism as a mechanism for regulation of immune responses. And in the last 10, 15 years, we have seen a lot of results coming from very good groups talking about the metabolic regulation of immune function. And what we have learned from these studies is that the function of effector cells is associated to cell metabolism, which is strictly linked to the metabolic status of the individual. Is it possible to study the metabolic pathways that support immune cell function in vitro as well as in vivo? And this led to the identification of pathways and enzymes that can be used to effectively modulate immune responses in vitro, but now also in vivo. And I will show you some data about this. But we don't know which is the effect of these pathways during aging. So today I will talk to you of the effects of aging on many of these metabolic pathways. And I will show you now some data on how aging changes the signature, the metabolic signature of an individual. So we did a small metabolomic analysis together with our Department of Pathology, and we analyzed 100 markers of inflammation and metabolites. And we look at triglycerides, LDL and HDL, glucose, albumin, alanine, transferase, creatinine, ferritin, C-reactive protein, serum amyloid A. This enzyme, very interesting to me, which is lactate dehydrogenase, and fatty acids. So these that I'm showing here are the only 12 out of 100 that came out significantly different in young and healthy and elderly healthy people. These are 20 per group, and the elderly are over 65 years of age. And in fact, when we do a PCA analysis, you can see that based on these 12 markers of inflammaging and metabolic dysfunction, we see the very separated population of young from elderly. And these actually are among the markers that have been used in a series of studies coordinated by the late Janet McElhaney in Canada to establish the biological age of individuals over 65 years of age. And why are these metabolic markers important? Because they can predict the capacity of an individual to respond to infections or vaccination. And this is an example, again, with a lactate dehydrogenase, which is an enzyme that I will talk to you later in my presentation. So we measured the antibody response in serum during the recent pandemic to the spike antigen of the SARS-CoV-2 virus, and we saw a significant difference between young and elderly individuals. But the most interesting thing is that the people that were unable to respond in terms of amount of antibodies, just the total amount in serum, were those that were expressing the higher levels of lactate dehydrogenase in serum. And this is true not only for this marker, also for many others. And the same is for the influenza vaccine response measured by the hemagglutination inhibition assay. So the people that respond less because they're old are the ones that express very high levels in serum of this lactate dehydrogenase enzyme. So we think that, of course, now we're putting together all the data, the biostatistical people are working on that, but actually it is possible to have a signature, very accurate signature of each individual, and this signature can predict the capacity to respond or not to a lot of exogenous stimuli. Now, the serum profile of the individual is associated with the intrinsic metabolic profile of the immune cells from this individual. And now I will show you some data obtained in healthy young and elderly individuals, as well as in elderly, same age and gender and everything matched, that are with type 2 diabetes, but they are naive for treatment. So they're not taking metformin. So this is just to show you from the results that I'm going to present that not only aging, but also any condition of disease which is inflammatory based on top of aging is deteriorating further the immune system. So we studied the metabolic phenotype profile of B cells. These are the immune cells that make antibodies. We studied these from our cohort of the 3 groups of individuals, and we did unstimulated because what is important is in the unstimulated T cells in order to predict the response of the same cells to in vivo or in vitro stimulation. And we use the seahorse and the mito stress test. This is a system, a tool that is allowing the real-time measure of 2 major pathways, glycolytic pathways in the cell. So one is called oxygen consumption rates, and is measuring the mitochondrial function of the cells. And the other one is called ECAR, extracellular acidification rates, which is the change in intracellular pH, which is due to lactate secretion. And this is a measure of anaerobic glycolysis. So we have mitochondria and we have anaerobic glycolysis measured at the same time in the same cells, loaded on the seahorse machine. And this is a typical profile of the oxygen consumption rates, which are mitochondria. This is basal respiration that is then inhibited by the addition of oligomycin, which blocks one of the 4 complexes of the electron transport chain. So we have basic level of respiration that can be increased by the exposure of the cells to an uncoupling agent. So this is the maximal capacity of the cell to do respiration, which is then blocked by these 2 compounds that shut down the system to basic level, showing that the respiration actually is mitochondrial. And this is what we have several measures, but here I'm showing you the maximal respiratory capacity in the 3 groups of individuals. We run only 5 out of 20 because the test is very expensive, and we don't have enough cells to do from all the individuals. These are B cells sorted from the peripheral blood. You see, aging is inducing an increase in maximal respiration, and type 2 diabetes on top of aging is inducing an even higher profile. Again, these type 2 patients are without metformin. Metformin is an anti-inflammatory drug, hypoglycemic and anti-inflammatory, so it's doing something, but not in all of them. And this is the spare respiratory capacity, another important measure. This is the difference between maximal and basal. So this is actually the capacity of the cells to start well and do even better when we add the uncoupling agent. And you see, we have significant differences between the 3 groups, but we have even better differences when we measure the anaerobic glycolysis, so this pathway through ECAR. You see the difference here in maximal respiration, and this is the ratio between the maximal respiration in OCR and in ECAR. Of course, it's decreased because we have more anaerobic glycolysis. So this means that the cells quickly shift from mitochondrial to anaerobic glycolysis when you block the respiration with oligomycin. And now I'm going to show you in detail the different pathways. So everything starts with glucose uptake. So we measure the glucose transporter, and you see that the type 2 diabetic B cells have a very spread expression of this transporter, which is the major one in circulating B cells. And we also have a measure of the actual glucose that is uptaken by these cells. And the 2 pathways are strictly correlated. I'm not showing here the correlation. So the oxygen consumption rates results and the ECAR rates are reflecting a higher expression of glucose transporter and a higher glucose uptake. This is the expression of the other glucose transporters, nothing relevant. Here we have a significant difference, but this is the glucose transporter which is important in tissues, not in the blood. And then when we measure the different pathways associated with OCR, with oxygen consumption rates, first of all, we measure what is happening in the mitochondria after the transformation, the conversion of pyruvate into ATP. And this occurs in the Krebs cycle. So these are the different pathways. This is the enzyme that actually converts pyruvate dehydrogenase X, is the enzyme that converts pyruvate in the cytoplasm and then is processed in the mitochondria to generate ATP. When this pathway is activated, we have secretion of cytosolic ROS, which is an inflammatory signal. This is measured by flow cytometry. And we have also the mitochondrial secretion of ROS, which is measured with this other fluorescent analog. So the pathways in the mitochondria are all upregulated in B cells from elderly, healthy, and as a matter of fact, elderly with type 2 diabetes. And also the pathways involved in the biogenesis. This is an enzyme that is responsible for the biogenesis of new mitochondria, which is also evident by the staining with the mitotracker. So this is a marker for morphology and abundance of mitochondria in the cell. And you see that all of them are upregulated, as well as the phosphorylation of this protein that is measuring the fitness of mitochondria. So AMP is the AMP activated kinase associated with mitochondrial function. But again, the major results, at least in this cohort, was with the lactate dehydrogenase A. This is the enzyme that allows pyruvate to be converted into lactate. So there's a change in the intracellular pH and a very inflammatory signal that starts to happen inside the cytosol. So this is the first measure, the enzyme, which is upregulated by aging and type 2D. And this is, as I said, this is measured in unstimulated B cells. Then the same B cells are activated for 2 days in vitro with CpG, is a mitogen for B cells, and the cells make a lot of lactate. Why is this important? Because lactate is a signal to make autoimmune antibodies. And here we measure anti-nuclear antibodies, and we measure anti-malondialdehyde. This is a product of lipid peroxidation, is a pathway that is active in aging and in age-associated conditions and diseases. And the B cells do so because they express the transcription factor for autoimmune antibody secretion. So TBET is activated and is a dose response curve, is activated in the 3 groups, but especially in the elderly, healthy, and in those with type 2 diabetes. And of course, lactate and the antibodies are strictly correlated. So lactate is a signal for autoimmune pathogenic antibody secretion, as well as inflammatory cytokines. So we have a lot of information. And again, my interest is on B cells and humoral immunity. But if we look at the CD4, CD8, NK, and monocytes from these individuals, we can have very similar pathways going on. So lactate is made mainly by B rather than CD4 and other cell types. And I will show you later this data. So far I showed you that people that are old with healthy aging, and as a matter of fact, with a disease associated with aging, have a very high metabolic inflammatory profile in serum. And this is translated into a higher intrinsic metabolic profile of the immune cells, especially B cells. Now, the next question is, are these B cells able to also convert other immune cell types into inflammatory cell types? And the answer is yes. So we did a transwell experiment. And we put at the bottom of the transwell B cells from young or elderly individuals, still this healthy cohort of individuals, no diseases associated. And then on top of the transwell, we put CD4 coming from a young individual. So there is no contact to contact. This is a transwell, only soluble factors in these conditions. After 24 hours of culture, we take out the CD4, we isolate RNA, and we measure the metabolic pathways that I showed you so far. So glucose transporter 1, lactate dehydrogenase, and pyruvate dehydrogenase. And the results here are shown as fold increase, fold change, compared to what is expressed in the CD4 alone. So this is the denominator, CD4 alone, from young, and then CD4 plus young B cells, CD4 plus old B cells. So you see immediately high glucose transporter 1 and high lactate dehydrogenase. There is also some significant change in this case, but this pathway is still not expressed at the top. And of course, because metabolism means inflammation, so higher metabolic profile means higher pro-inflammatory profile, we measure two inflammatory cytokines that come from T cells. So we measure IL-17 and interferon gamma. Again, this is the ratio versus the CD4 alone. And you see we have significant higher levels of IL-17 and interferon gamma measured by flow cytometry after 48 hours of culture. So this is the supernatant here analyzed. And in these conditions, guess what? There is a lot of lactate. And this lactate comes only from the B cells, because T cells, CD4, don't make lactate if they are not heavily stimulated with nitrogens. So in these conditions, there is no stimulus. It's only what comes from the B cells and what comes from the CD4. So no lactate from the CD4. And this amount is much lower than the one that we have seen before in the stimulated B cells. But anyway, they make it even if they are in a non-stimulated condition. And what is lactate doing? Is activating NF-κB. NF-κB is the transcription factor for inflammatory cytokine secretion. So you make IL-17 and you make interferon gamma and many other, and IL-6, TNF, and many other inflammatory cytokines in the CD4 or in inflammatory cells, only if you have activation of NF-κB. So we took the proteins from CD4, and we made cytoplasmic and nuclear proteins from this CD4. So the activation of NF-κB is when the protein translocates from the cytoplasm into the nucleus. So in the presence of young B cells, there is not so much translocation. In the presence of old B cells, you see that the majority of NF-κB is in the nucleus, which means that it is going there, is translocated, is activated, it binds the promoter region of IL-17 and interferon gamma, and many other inflammatory signals. And of course, upstream of this, upstream of this NF-κB activation, there is STAT3 phosphorylation still in the CD4 T cells without stimulus. Again, the only stimulus is coming from either the young or old B cells, okay, without any additional stimulus. So lactate from B cells is inducing inflammatory CD4 from naive CD4. And I can tell you also CD8 and NK. No effects on monocytes, because monocytes are already hypermetabolic, so there's nothing that changed their profile. They're there, they're happy, and they don't change absolutely in the presence of any exogenous stimulus. So the next question is it possible to block this pathway? And the answer is yes, and we used actually 3 different inhibitors of lactate. One is this small molecule, FX11, this is the name, unpronounceable, that blocks the enzyme. So in the presence of FX11, the enzyme is not converting pyruvate into lactate. And when we pre-treated the B cells from elderly only in these experiments with this inhibitor, we have a significant reduction in the cytokine secretion. The same happens with this other one. This is an antibody that blocks lactate transporter. And lactate transporter is actually inducing a cycle of activation of lactate. So more lactate is made, more transporter is associated with. The two interact and generate more lactate secretion, which upregulates the lactate transporter. And this is a vicious cycle until lactate is completely shut down. It's shut down pretty fast in the cells because it's very toxic and is inducing apoptosis, is inducing a lot of cell transformation. So it's something that in maybe 48 hours, no later than 48 hours, you can measure lactate, but only the metabolites, which is something that we are now going to analyze using mass spec. So this is a very effective block of IL-17 and interferon gamma using the antibody that blocks the lactate transporter. And then there is another thing that I am very interested in, and is the blocking, which is relevant, of course, more in vivo than in vitro, and is relevant, for example, in the tissues. Because this is a pathway, ICOS ligand. ICOS ligand is expressed by the B cells, and in vivo they interact with CD4 that express ICOS. And this is metabolic, is glycolytically supported. So if you block glycolysis, and if you block any one of these pathways, you don't have the interaction in vitro between B and T cells that express this cognate way of interact. And of course, with these three kind of block, we also block the amount of lactate secreted in the co-cultures. So everything makes sense. Now, I will focus on lactate because lactate is not only relevant in these conditions, cultures and co-culture, but is very relevant in many conditions associated with aging. And aging actually induces an increase of this metabolite, which is mainly made out of the pyruvate metabolism and in muscle. But immune cells in blood make a lot of lactate. That was kind of unexpected, at least to me. Then you have it in tissues, you have it everywhere. And the amount, again, the amount of lactate that is circulating is kept under control, is immediately degraded, made and degraded, because if not, is very pathogenic, is very toxic. So lactate, as I showed you before, is made after stimulation of B cells from people with healthy aging. This is what we have experienced with type 2 diabetes, people with obesity, people all elderly with rheumatoid arthritis and other autoimmune diseases, as well as breast cancer. You see that everything is upregulated as compared to the healthy aging. And when we look at the synovium of these individuals with RA, there is even more made in the synovial fluid, and is very pathogenic and very dangerous there. So the immunometabolic effects of lactate are known for many immune cell types: T cells, mast cells, MDSCs, neutrophils, but not for B cells. So we studied this effect by taking B cells from young individuals and put them in culture overnight with or without lactate. And then we did this in the seahorse, but using a glycolytic test. So in this test, we induce the maximal activation with glucose. Then we block the mitochondrial respiration with oligomycin. And then, of course, everything that is glycolytic from this point on is upregulated. And then the reaction is finally stopped by a glucose analog, the 2-deoxyglucose. So when we did this, we saw immediately that the presence of lactate is inducing this inflammatory and hypermetabolic phenotype of the B cells, which is also parallel by an increase in the expression of transcripts for glucose transporter 1, of course, lactate dehydrogenase, and much less for pyruvate dehydrogenase. But when we put in the picture the B cells from elderly individuals untreated with lactate, we have an overlap of these two groups. So actually lactate is inducing a status of immunosenescence in B cells from young individuals. And also the parameters here the metabolic parameters are absolutely overlapping between the young with lactate and the elderly without lactate. So is actually favoring an immunosenescent status. In these conditions, there are a lot of markers associated with the senescence-associated secretory phenotype, including the markers of senescence P16 and P21. And these cells can be positively labeled with beta-galactosidase, which is a marker of immunosenescence in vitro. a lot in tissues, but also in blood. So these cells are becoming senescent. And after 24 hours or 48 hours, you can actually see an increase in the frequency of cells that express this marker of senescence when the young B cells are in the presence of lactate, similar to what happens with the elderly. And also the intensity of fluorescence per cell is upregulated. So it's actually inducing markers of immunosenescence. And of course, this is associated with the secretion of IL-6 and TNF-alpha, inflammatory cytokines, as well as of autoimmune antibodies. Okay, so lactate induces immunosenescence, inflammation, and pathogenic B cells. And again, this is possible because the B cells express large amount of the lactate transporter, this SLC5812. And they have it, of course, among the lymphocytes. They are the ones expressing the highest level as compared to CD4. And then you have CD8 and K and other cell types. Of course, less than the monocytes. These are the highest in these pathways. And of course, the expression is much higher in the elderly as compared to the young, threefold more at least, but the pathway of expression is very similar. And as I showed you before, this is the upregulation of the lactate transporter when you expose the cells to lactate. And this is similar to what we have with the elderly. So lactate is upregulating the lactate transporter, which is inducing more lactate secretion in this vicious cycle of inflammation. And of course, we can use the antibody that I used before in the co-culture settings to downregulate the expression of the transporter. So this is very specific. And in these conditions, the antibody is also blocking lactate, is also blocking the secretion of pro-inflammatory cytokines and pathogenic antibodies, either in the young exposed to lactate or in the elderly without any lactate. So which means that there is a spontaneous upregulation of lactate secretion in the elderly without any addition of lactate. Now, this is due mainly to the redistribution of B cell subsets with aging. We have several changing in B cells as well as CD4, CD8, and NK with aging, and we have an upregulation of the pro-inflammatory cell types when the B cells come from elderly individuals. This is the flow cytometry to show our gates. These are the lymphocyte, the CD19 positive, and then from here we use the CD27 and IgD markers to evaluate the four major subsets of circulating B cells, which are the naive, IgM memory, switch memory, and double negative. So the major changes are at the level of these two populations. The double negative that go significantly up with aging, and the naive that also go up with aging. So which is of these two populations responsible of all the effects that I showed you with lactate? Maybe both of them, because the other two don't seem to have big effects or significant effects. So we did this many years ago, and now we have additional data on the sorted population. But what we saw in the past is that only memory, so double negative, switch memory, and IgM memory, but not naive, are able to secrete inflammatory cytokines and chemokines, markers of the SASP, or inflammatory microRNAs. And among the memory, the double negative are the most inflammatory B cell subset. So everything seems to come out of this population and not of naive, that also increase with age. So naive are not inflammatory, double negative are. And these double negative here are actually in the previous one, this population is the one that increases significantly in all the inflammatory conditions associated with aging. So type 2 diabetes, obesity, autoimmune diseases, cardiovascular diseases. In all these conditions, this population takes the lead. So again, with the PCA, you see that the double negative are separated from the other population, but they're very separated from the naive. So no contribution of naive B cells to what we have seen so far. And of course, these double negative are metabolically supported. This is something that in 2017 we didn't look at, but now we have a series of experiments with fluidyme and now also single-cell RNA-seq, showing that not only the lactate the glucose transporter 1 and the lactate dehydrogenase are upregulated, but a lot of different glycolytic pathways that come from glucose and go into pyruvate and then in this pathway. So this B cell subset is glycolytically supported, which is the pathway that generates pathogenic antibodies and inflammatory cytokines. So at this point, I want to summarize what I showed you so far, that aging changes the metabolic serum, the systemic metabolic profile of an individual, and inducing hypermetabolic immune cells, B cells, but not only. These B cells are able to polarize naive CD4 or other cell types into inflammatory T cells. And also that aging induces secretion of lactate, and lactate is a signal for immunosenescence. And the majority of the changes that we see in the total B cell pool with age reflects changes in the double negative rather than in naive, which also increase with age. So these data are in the medical repository publication, and they are being resubmitted after the second review to Nature Communication. So I hope it will be accepted soon. So now finally, I showed you some intervention in vitro with pathways that block lactate secretion. And there is actually the lactate transporter 1 that is now in clinical trials. There are some small molecules as well as different antibodies that block that pathway, but the results are not available yet. On the opposite side, there are some clinical trials with senolytics. And these senolytics are very effective in reducing the burden of senescent cells in tissues, in the circulation as well as in tissue. And because I work a lot with the adipose tissue, I will show you now some results from the human obese adipose tissue treated in vitro with these two senolytics, dasatinib and quercetin, that are very effective in, again, in reducing, not resolving the problems associated with obesity, but reducing the charge of senescent cells in the tissue. So we have access to large amounts of discarded adipose tissue that comes from weight reduction surgeries. This is breast. And then we do the collagenase digestion, and we have immune cells. Every kind of immune cell is present in this stromal vascular fraction. It's a real immune tissue, the adipose tissue. It's not only a storage of nutrients. And then we also have some adipocytes that have a different profile from the immune cells. So what we did was, first of all, to stain the stromal vascular fraction, or SVF, after an overnight incubation with the two senolytics together, and by flow cytometry looking at beta-galactosidase, which is, again, a measure of senescent cells. And you see that the frequency of cells is decreased. This is done by flow cytometry gating on leukocytes and 19. So this is a measure of the B cell beta-galactosidase inside the tissue. And you see that here, in the presence of the two senolytics, the MFI of the beta-galactosidase is decreased. And these are the data from the 6 independent individuals. So very effective in reducing the senescent markers. And these two have been used in vivo in people taking the senolytics for 14 days. These are drugs that have to be taken and then suspended and then taken again. And the analysis made by Kirkland and the Rochester group is on the small biopsies before and at the end of the treatment, small biopsies from the obese tides of these obese patients, looking at the amount of beta-gal positive cells. So reducing beta-gal positive cells, we also reduce other markers of the SASP, P16 and P21, as well as the IL-6 and TNF. This is done on B cells sorted from the stromal vascular fraction. And of course, reducing the amount of senescent cells and of senolytic and SAS marker, we have also a significant reduction in the metabolic pathways that support secretion of inflammatory cytokines and pathogenic antibodies. We are now measuring the antibodies. And this is my last, my final result slide. Again, I want to thank the people that did the experiments. My doctors, so important when you want to do some study on human people, to have them on your side and to support and be really involved and participate in the research. My longtime friend and collaborator, Valkyria, the fundings, and of course, all of you that are here today, as well as those that are in the air. Thank you so much.
Thank you, Daniela, for this very nice presentation. I will open for questions, so if anyone would like to ask something, Gisele, please.
Thank you, Daniela, for the talk. I would like to know what you're thinking about the clinical application of these inhibitors of lactate in terms of old patients and the impact, for example, for vaccination effectivity or things like that.
Well, that's a very interesting point. So from a clinical point of view, not vaccine related, there is a reduced, this is what is expected from the clinical trial, a reduced burden of morbidities and comorbidities, because you reduce a stimulus for inflammation that is associated with these conditions. For a vaccination point of view, the people that have very little amount of lactate in the circulation are those that respond better. I showed you the influenza vaccine. The COVID vaccine is exactly the same. We did a study with both Pfizer and Moderna vaccine. I mean, the results are really black and white. So lower is the serum level, and of course, the intrinsic B cell level. We look also at the P of the circulating follicular helper cells, and of course, the response is greatly enhanced. Yeah.
Thank you.
Professor Daniela, thank you for the great talk. It's amazing to see how lactate can stimulate our immune system in the bad way. And it's interesting because it seems to be an anaerobic condition that you drift part of it from the Krebs cycle, and then you produce a lot of lactate and induce those inflammatory cells. My question is about the changes in B cell subtypes, because I expected to be to have an exhaustion from the bone marrow, and you would have much more, how could I say, the memory phenotypes than the naive, as it happens in T cells, because in T cells you see less naive cells and more central memory and effective memory T cells. So they do not contribute to the aging, but do they respond well to new antigens? Because in the T subsets, we don't see that response because of the thymic atrophy with aging.
Very, very interesting point. Thank you for being here again. And for the circulating B cells, there are no exhausted B cells that we can stain. So we did the PDL, PDL1, you know, these cells, even if they express a little bit of these markers, they are still highly secretory, so they are not exhausted. Conversely, if we look at the tissue, the profile of the subsets is totally different. no naive, only memory. And this is because in the tissue, and when I'm talking of tissue, I'm talking about the obese adipose tissue, and I'm talking about the synovial fluid from RA patients, because this is a continuous status of activation that is inducing continuous chronic class switch. So you have a reduction in naive because these cells go into the pathway of IgM memory, switch memory, and double negative, and then they accumulate. But again, for the B cells, I don't see so many exhaustion because even the double negative, which represent the terminally differentiated subset, is still metabolically and transcriptionally very active, which is a condition that is not associated with cell exhaustion, at least for a prolonged time. For the T cells, the situation is different. And this is different also in blood. So you arrive to a certain point where these TEMBRA, or terminally differentiated T cells, are not secreting or doing anything more over a certain point. So the two immune cell subsets are different. And again, for B cells, blood and tissue are showing significant differences.
Oh, great. In the past, I thought that M1 macrophages were mostly responsible for the inflamed aging, but other cells and the double negative B cells are very reactive and produce a lot of pro-inflammatory cytokines.
That's why in the early 2000s, together with inflammaging, we saw the term macrophaging, but actually not only B cells. So there are a lot of other cell types that are, and then again, in the tissues, you have a continuous transformation and polarization of generating only M1 macrophages, because you start from non-inflammatory and then for the stimuli that are secreted constantly. And I think that there are, at least these are preliminary data that we have, that some metabolites of the lactate are having better effects than others. And this will be the next step to evaluate. So which are the most effective in generating the pathogenicity of the B cells? And are they working the same in B and T? I don't know.
Great. The last question, a person that sums up rheumatoid arthritis, obesity, diabetes, CMV, chronic carriage, so all in one person. So that means that the immune system is almost kicking the bucket. I mean, at the end of the immunosenescence with several stimulating conditions.
Well, in the case of rheumatoid arthritis, at least in our cohort of people that have or not obesity, there is a different response to therapy, but they respond. So I like to see before and after therapy, and I showed on Friday the data without any treatment, as well as these ones. And this is what is making me more interesting, to see how is the steady state condition of the person. But they are responding, especially those with lower levels of obesity, they're responding pretty well to treatments. And these biological agents are kind of not flipping completely, but inducing, again, protective responses to vaccination. So actually we have a very good COVID and HAI response to the influenza vaccine. So this is just the cohorts without any treatment. But treatment that are tailored in the most cases of the patients are showing beneficial effects. So it's not that they don't respond, they die because they get infections. No, actually the disease that is inducing all these bad things can be kept under control in a significant way. I have this data, but too much to present.
So, Daniela, I would like to ask you if the lactate production has any relation with the glycemic level of the person you have?
It does, actually. I don't have these results because we only measure how much is in serum and how much is in the immune cells. And again, has to be kept under control. So what is in serum actually is a reflection of secretion and degradation, secretion and degradation. It's more accurate, the one that we make in the cells. It's pretty stable over time, like 2-3 days that you measure on culture supernatant stay the same. In serum, it depends a lot from many other conditions. So it's made, and that's why we don't have any measure of lactate in serum, just the enzyme. And I hope we will have interesting results measuring the metabolites.
Yeah, but I mean, the results you got on lactate on in vitro, do they correlate with the in vivo levels of glycemia? You
Of
know?
glycemia. No, I understood a totally different thing. Yes, they do. Yes, they do. We measure glucose. Yeah, they do.
And
I thought about lactylation. No, it's a different thing.
And does lactate has any relation with the gender? I mean, you can get different results if you're studying males or females.
I haven't seen so far talking about the lactate dehydrogenase in serum. We have 10 and 10 males and females, and I haven't seen differences. Where I see differences in the amount of autoimmune antibodies. So probably females have more. So that's the only measure where I see, and IL-6, that's the other measure that is favoring that is higher in females. But actually on 20 people, we need to have more in order to have a very good correlation that doesn't have false positives or negatives. So actually not. For lactate dehydrogenase, the values are similar. I don't know, we have to measure the lactate metabolites, and then I can answer better this question.
So let me ask you one last question. I have heard so many times about quercetin, not only as a senolytic drug, but also in cancer and other chronic diseases, but never was really used as a treatment. So do you think that now you're getting these positive results as senolytics? They will start like really using quercetin? Maybe I'm wrong, but I always seen quercetin.
I am not the right person to answer this question. I know that in Rochester, at the Mayo Clinic, they are the leaders for the senolytics, and they are using it together. Dasatinib and quercetin work when they are together. They don't get very important results using one alone. I didn't try because that experiment with the adipose tissue was done together with Christina Kamel. Was not done together, but was suggested by experiments that she has done at the Mayo Clinic. And Christina Kamel comes from Yale, so she has a very good background on adipose tissue. So I know that they have used them together because the effects are not so significant when used alone. But they're still in clinical trial as a single molecule, either one or the other, including quercetin. Okay.
No more questions? No? Thank you very much,
Thank you.
Daniela. So we have the next speaker, Professor Gisele from the Geriatrics and Gerontology. She's gonna come in front here and talk to us. Thank you very much, Gisele, for accepting this invitation.
Thank you, Valkyria, for this invitation. In fact, this is the second time that I come to this CAPES PrInt meeting. The first time was still working as a hematology oncologist 5 years ago. And during this meeting, it was full of people here from many areas of aging. I realized that it was an area that I would like to study more. So I thank Dr. Valkyria for my shift career at this moment, because 2 years ago I decided that I would like to study aging, because aging is one of the most important factors related to cancer development. And this is the one that we cannot change. But many other related to lifestyle, we can change. So this is what I'm trying to do in geriatrics currently. And I believe that we have a good field to work, avoiding many diseases that affect old patients like cancer. So I believe that the answer, it's not only the treatment, but the prevention. So that's what I'm now trying to work with aging. So my background is as a clinician, and also in cellular and molecular biology. So 2 years ago, when I started in outpatient service in geriatrics, I'm trying to understand what they are doing there. So I need to learn everything about clinics, and also about molecular and cellular biology in aging. So I started with something that was a little bit more familiar to me, that was hallmarks of aging, because they share many components that are now known that hallmarks of cancer too. So I think that starting from hallmarks of aging, it would be easier for me. So here we have a lot of markers that are well known, but they are totally connected. And more recently, we have updated of those hallmarks, including, for example, microbiota and inflammaging as relevant biomarkers too. But I started with the initial hallmarks that were published in 2013. And you see here cellular senescence that Dr. Daniela spoke to us today, and this is the interest of Dr. Valkyria too. And Dr. Maisa Sendoroglu, that is the responsible for the outpatient service where I am working now, she was also interested in cellular senescence. But I felt that working with senolytics will be very difficult to us. Dasatinib is very expensive, is oncologic drug too, and here in Brazil we don't have even this available for cancer patients that need dasatinib. So it was very difficult to work with this in clinical practice. So I decided to move to other hallmark that should be easier for us. And it was the regulated nutrient sensing. Nutrient sensing is a cell ability to recognize and respond to fuel substrates such as glucose. And insulin resistant type 2 diabetes, obesity, and metabolic syndrome are consequences of this dysregulated nutrient sensing. And here, this is a very busy slide, but we have the 4 pathways of nutrient sensing to regulate metabolism and influence aging. The 4 elements are insulin and insulin growth factor 1, that it's here, mTOR, citrulline, and AMP kinase that Dr. Daniela talked to us, that is very important in this pathway too. In this figure, you can see that caloric restriction continuously or as intermittent fasting is able to promote a lot of anti-aging effects, like increased DNA repair, increased autophagy, detoxification, neurotrophic hormones, avoiding, for example, loss of cognition, reduce apoptosis, reduce senescence, glycolysis, and inflammation, like Dr. Daniela showed us. So this is one of the most important interventions that we can do. It's not very difficult to have anti-aging effects that are very, very important. But looking at this figure, we also see that there is a drug that is relatively cheap and accessible, even here in Brazil, that is metformin. And metformin decrease mTOR and increase AMP kinase. So during these two mechanisms, metformin has a very interesting anti-aging effect. So that's why we decided to start from metformin and see what we can find. Working with Dr. Maísa, we are part of this Longevos project. There is a project that started in 2010, taking care of 80 plus years old patients that enter the project in a very independent way. It means that they are able to do all the basic daily life activities and also instrumental activities of daily life. Basic daily life activities are like eat alone, dress up alone, have a shower alone, and instrumental activities are deal with money, shopping, make his or her own food. So they are a little more complex. So all the patients that enter this project are independent in both aspects, and they are follow-up during 13 to 40 years now. And working with those patients, I found that there are two major problems that are developing during this aging process: dementia and sarcopenia. And thinking about these two major problems, I decided to investigate if metformin could change the prevalence of dementia and sarcopenia in this group of patients. But we are able to prescribe metformin only for diabetic patients. So diabetic patients are our goal. We should study diabetic patients and see if they behave in a different way comparing to other patients that are 80 plus and not having diabetes. So this is the first paper that I showed you, and most of the work that I will present today is done by Maísa Aguiar, that is now, she was our previous resident, but now she's a PhD candidate, and Solomon King, one of our med students here in Unifesp. And so they collected and analyzed the data that I will present to you. This is a very simple study, and the other that I showed you too, but it was our beginning. I was learning everything in geriatrics, and we have to have a start. So this is our start. If you take a look at this very busy figure, you see that metformin has very important action in improving cognition. Metformin is able to promote cell survival and neurodifferentiation, increase autophagy, improves mitochondrial biogenesis. It causes neuroprotection, neurodifferentiation, proliferation and self-renew of microglia. So there are very, very important actions that could be used in favor of metformin to improve cognition. And many studies have shown now that metformin may reduce the incidence of dementia, suggesting a geroprotective action. So, as it is an accessible drug, we asked if we could compare the prevalence of dementia among long-lived people with and without diabetes, using or not metformin monitoring in this Longevous project. So this is only observational study. We did not do any intervention. And in the first moment, we start evaluating 194 independent patients over 80 years old, follow up for 13 years. It was a case control study. They were divided in non-diabetics and diabetics, and the diabetics patients in using metformin and not using metformin. So we collected data about functionality, cognition, some laboratorial tests, among others. And we made the collection of data from the first consultation. It was relevant for calculation of overall survival and dementia-free survival, but all the analyzed data that I showed you were done in the last evaluation. Variables were analyzed by standard statistics and also the survival curves. So we found that this population was composed mostly by women, as expected after 80 years old. The median age of this group was 90 years old, varying from 80 to 106. The median follow-up time until death or a major outcome like infarct, vascular cerebral accident or cancer that makes the patient dependent. So in this situation, they have to be removed from the study. So the median follow-up time was 4 years, vary from 0 to 13 years. And the prevalence of diabetes in this group was around 25%. What makes our case control study like 3 to 1, or 1 case to 3 controls, okay? From the diabetic patients, half of them were using metformin, a little bit more than half of them. Well, this is the first form that we analyzed the group, comparing 3 controls for each case, and all of them were compared using those variables, like age, like schooling, like fast blood glucose and glycate hemoglobin, basic activities of daily life, instrumental activities of daily life, depression using a geriatric scale, and a mini mental examination test to see how the cognition of those patients is. So one thing that calls our attention in this first table is both groups are completely compared for all those parameters, excluding fast blood glucose and glycate hemoglobin, of course. And they are still in the end of the follow-up, independent for basic and instrumental activity of daily life. The dementia prevalence among all patients was around 32%. So this is a data that showed that called our attention because we didn't have this number previously. So even with starting with in this independent situation, during the follow-up, 32.6% of the patients developed dementia. So this is a huge problem. If you can take a look in the second table, we see that the prevalence of dementia in diabetic patients is a little inferior if you compare with the rest of the population, but the difference was not significant, okay? Comparing non-diabetic patients with diabetic patients using metformin, we did not find any difference except with the glucose levels and hemoglobin glycate levels. And the prevalence of dementia between the two groups was not significant. But again, patients with metformin have a little lower incidence of dementia compared with the rest of the population, but it was not significant. Comparing diabetic patients based on metformin use, patients with and without metformin, we did not find any difference among the parameters and also with the incidence of dementia. And comparing non-diabetic patients with diabetic using metformin, we did find any difference, and here dementia was not significant, showing that metformin did not make difference comparing even controls and diabetic not using metformin. Taking a look on survival course, we did not find any difference between groups, and the first curve shows diabetic and non-diabetic patients. It was the same behavior. And here, the most important for us, diabetic patients taking metformin and non-diabetic patients, no difference. So we move to probability of disease-free survival, probability of dementia among those patients, and we did not see any difference between diabetic and non-diabetic patients. And comparing non-diabetic and diabetic taking metformin, the difference was not significant, but after 5 years of follow-up, you see that the curves are trying to separate with better behavior for the metformin users, but still it's not significant. So here we can conclude that as far as we know, this is a pioneer study in evaluating the prevalence of dementia in diabetic and non-diabetic patients aged over 80 years old living in the community. Metformin probably was not protective for those patients, and we believe that the adequate glycemic control among diabetic patients is responsible for the absence of this difference. Independent of the treatment that the diabetic patients are taking, they are very, very well controlled. So probably they are not suffering from dementia more than it was expected if they were not diabetic. And this is very relevant because diabetes is the most well-established modified risk factor for the development of dementia among the elderly. So we don't need only metformin, we need the adequate control. So at least for this patient, 80 plus years old that started the project in a very independent way, we know now that the development of dementia for the whole group is 32.6%, independent on being or not being diabetic, using or not metformin. Okay, so we are not taking metformin since tomorrow because we want to have all these benefits of metformin avoiding dementia. At least in this group of patients, we cannot say this. We have to continue to control the diabetics as well as it has been doing for this group. Our second question is if metformin should be important for the sarcopenia development. And again, Maisa and Solomon collected all this data, and Maisa made all the statistic analysis with our statistician, Dr. Adriana Bruscato. So we can say that sarcopenia is the loss of muscle mass and the function, and is a very common condition in the elderly, especially in diabetic patients. But metformin can be a geroprotective drug. But there are many studies that say that it can contribute to muscle loss. So, is metformin protective or deleterious for diabetic muscle mass? That was our question. So the objectives of the study was to compare the prevalence of sarcopenia among elderly people treated for diabetes, case group, with or without metformin, and non-diabetic patients monitored in the Longevous project, and also to evaluate if there is an association between the use of metformin and the development of sarcopenia in this group of patients. So this is the same patients, okay, the same 194 independent elderly people, the same almost 25% of diabetic patients, half of them using metformin. But here we have some particularities. Sarcopenia was defined using international guidelines, but we used hand grip, we use the calf circumference, and we use the gait speed to define sarcopenia in those patients. Here in Brazil, it's not easy to measure the muscle mass in a hospital like ours, that is a public hospital, because the measurement of the muscle mass depends on bioimpedancia or CT scans or DEXA. They are very difficult to do in our practice. So calf circumference is a way that is validated in many populations now, and here in Brazil too, for younger and older patients. So it's okay because we have enough literature to use calf circumference for this purpose, and also the international guidelines approve the use of this measurement for people that do not have access to more expensive protocols. So the statistics was the usual one. And here we have again the table showing that comparing cases and controls, one case for 3 controls, there was no difference among the variables here, except fasting blood glucose and glycate hemoglobin. And here there are the parameters for sarcopenia, like calf circumference, hand grip, and so on, gait speed. And the sarcopenia prevalence among those patients was 16%. And 6% of the whole group have severe sarcopenia. And we didn't have this number previously. So now we know that sarcopenia is a problem, but not as prevalent as dementia for this population. Comparing non-diabetic patients with diabetic patients using metformin, we did not see any difference in the parameters, and also in the development of sarcopenia or severe sarcopenia, or even death. Comparing diabetic patients based on metformin use with or without metformin, we did not see any difference in the parameters, and also in the development of sarcopenia, severe sarcopenia, or death. The same overall survival curves that I presented before, because it's the same group, no difference between non-diabetic and diabetic taking metformin. And the probability of survival for sarcopenic and non-sarcopenic patients was not significant too. And also for sarcopenic and severe sarcopenic, the difference was not significant. So one possible limitation of the study is the use of calf circumference. We know that when we submit the paper, probably it will be refused because of this parameter that we used, despite of a lot of literature that support the use of the simple measurement for the sarcopenia definition, the muscle mass definition. And it was not possible to analyze sarcopenia-free survival because we don't have this data available. So our conclusion is that long-lived people with well-controlled diabetes did not show significant differences in relation to those without diabetes for the outcome of sarcopenia or death. And metformin did not have a negative impact on the progression of sarcopenia or death in this cohort. So all of our clinical colleagues said, oh, you will find a lot of sarcopenia among the diabetic patients because we see this every day. In this population, we did not see this. So we feel very, very happy with this information. Probably metformin has not a protective effect, but there is no deleterious effect. Well, so we are a little disappointed because we believe that we'll have a very good drug that will start prescribing metformin from everybody in this outpatient service, but it's not true. And metformin has all of these important effects on brain, on muscle, and even in gut. And trying to link all of those elements, we have this gut-brain-muscle axis that is very important now for many specialties like psychiatry, or even for those who work with sports medicine, because we know that everything is connected. So we have this data and we are so disappointed about our results. And I asked Maisa to take a look if we could correlate the muscle variables with the brain variables and see if we find something. So all of those analyses were done, and I will show only one graph for you to take a look what Maisa found. So here we have hand grip, and here we have the mini-mental state examination, and we see that there is a correlation. P was significant. But when we take a look in the Spearman coefficient, we see that the relationship is weak. So we can see also in this heat map that hand grip was correlated with basic activity of daily life, instrumental activity of daily life, but also as happened with the mini-mental, the association was weak, even with significant values of P. Then we decided to talk with our statistician to see if there is a more elegant way to take a look on those associations, because hand grip seems to be an important tool to evaluate cognition and mental status and functioning in those patients, because these three variables are most related with cognition than to physical performance. So Dr. Adriana told us that the linear regression should be an option, and in the linear regression we should do this thing. We'll use the hand grip, that is the strength in the hand, the right hand or the dominant hand of the patient, as an independent variable. And we use all the others as a dependent variable. And if the regression was, the linear regression was positive, we can say that hand grip could preview the other variables, and we are interested in the cognition related ones. So we saw that hand grip was associated with instrumental activity of daily life. We can see here this association, the P was significant, and it means that the hand grip, the strength in the dominant hand, that is a physical performance marker, was related with very complex activities that demand a lot of cognition from the patient. So this is a very interesting finding It's not new. There are few manuscripts in the last 5 years about this subject, and we are asking if we will still pursuing other markers, because currently we have 420 patients in this project, and here we analyzed more or less 200. So we can double these casuistics and see if we can find something in this gut-brain axis. And the other step that we would like to take next is the analysis of one pathway that can correlate the 3 elements of the gut-brain-muscle axis. That is the tryptophan metabolism. Tryptophan is an essential amino acid that is taken from diet, most from protein derivatives like meat and milk. And 5% of all tryptophan that we take is used to produce serotonin and melatonin. It's very important for many neuronal functions. And 90% of the tryptophan is metabolized in the liver, and some tryptophan that persists in the gut is metabolized also in the kynurenine pathway. And the kynurenine pathway produces a lot of products that are very toxic for the central nervous system. So here, maybe we have a pathway that we could manipulate with inhibitors to improve at least dementia in those patients. So our next goal is to study patients with dementia, sarcopenia, patients without dementia, without sarcopenia, and see how the tryptophan pathway behave. It's not easy to do. It depends on mass spectrometry, and it's very expensive. But there are many ELISA kits that are available currently, and they are cheaper if you compare with mass spectrometry, and easy to reproduce even in laboratories that are not that sophisticated. So if you would like to have a good biomarker, we have to have one that everybody can access and not only use mass spectrometry. So this is probably will be our next step, trying to investigate this pathway. And the originality is the comparison with dementia and sarcopenia, because all the studies show only one of those aspects, and we ought to put everybody together, because we really believe that everything that happens in the gut is related with the brain and the muscle, and we can see, as we see in geriatrics, the patient as a whole, not only as brain or muscle or gut. And this is the very beginning of my new career in geriatrics, with the help of this very talented students, that Maisa Aguiar, that now is a PhD candidate, Solomon King, that is also our med student, now in the 5th grade. And I would like to thank Dr. Maisa Sendorolo for all the things that she is doing for me in this new phase of my life, and Dr. Adriana Bruscato for support our statistics analysis. So thank you very much for the opportunity, and I hope that in the future I have more laboratorial data to show you. Thank you.
Thank you, Dr. Gisele, for this nice presentation. So we are open for questions. Yeah.
Congratulations, very, very interesting data. I have, let's say, a comment on the population, because you showed an average age of 92. So these are approaching the centenarian age. So maybe the fact that you don't see the effects of metformin, compare the diabetic with metformin compared to the normal controls, can be because they are already in a, let's say, pretty good quality of life. And this is shown by your values of the glycated hemoglobin. These are values of healthy and centenarian people. So just a comment, what do you think?
Yeah, one thing that is, this is a very particular population because now we are receiving patients from many other outpatient service in this institution. And if you compare how those patients that are very well controlled for those years, 13, 14 years now, and a patient from the same age that comes from another clinics, they are completely different. You cannot say that they have the same age. Because the patients that are well followed by geriatrics, they are seen as a whole, not only as a part of the body. And if you receive patients from oncology, neurology, rheumatology, the patient comes with dementia, but has a well osteoporosis control, for example. The patient comes with many cardiovascular alterations, but comes from neurology, so the neurologic part, it's okay. So they are in a very bad situation. So we decided that we have to compare these two populations because this population from the Longevos project is very special. This is not our reality. And the patients that come from the whole hospital, this is reflect how bad we are treating our old people. This is true. But the good thing is, after the geriatrics analysis, all the problems is, they want to solve the major problems first, but after a few months, they are doing better. So they are completely different. This approach, when you have only a specialists looking at those patients, like 3-4 specialists, different specialists looking at the same patient, the approach is completely different from the geriatrics group. So I believe that this group that comes from the other clinics reflects more what is happening with these old patients than the people from Dr. Maisa project. They are very well followed, so they are different. I believe that you are right. But they are different, but they have a lot of diagnostics. But they are compensated. They have hypertension, they have diabetics, they have sarcopenia, they have osteoporosis and many other diseases, but they are all compensated. So the quality of life is better. I saw much more deaths dealing with oncologic patients than working with this 80+ population.
And I don't remember, you may have said this, is the population analyzed in a whole, or males and females have different outcomes?
We analyze it as a whole. 1/3 is male, 2/3 is female, but I believe that Maisa did it, and we didn't see any difference.
A general question, which are the target cells in the brain, the metformin target cells in the brain? Is the glia or
mostly the glia?
Yeah, mostly the glia. And what is metformin doing from studies in vitro? Is it known, right?
Yeah, I will show again this picture. I don't know if it's too small to take a look again, but in glia, the effects are very, very important in neurodifferentiation, neuroprotection, self-renew, autophagia, and mitochondrial biogenesis. All the green ones are the positive effects.
Thank you.
Morning, and thank you for your great talk. I'm also an epidemiologist and a nutritionist, and I have my comment is in the same line of Professor Daniela, that I have like some studies, like my studies or other researchers, and we usually see that first we don't have a good measure for muscle mass in epidemiologic scenarios like hospitals or free living older adults. So I agree with you with this, like cough circumference can be a marker, but only for when we talk about severe sarcopenia. And these numbers of 33 and 34, like for cutoffs, are not great for severe sarcopenia, especially in centenarian, almost centenarians,
Mhm.
right? So I was taking a look in the Professor Gonzalez paper that they tried to correct for obesity, and they have 32 and 34 for males, I think, and 31 and 32 for females. And before, in back in the 90s or 2000s, we usually used this cutoff of 31 in hospitals, like
Mhm.
from the mini nutritional assessment. So probably for this particular group, we may be trying to put like more than one cutoff, to see like if we have a severe sarcopenia or mild sarcopenia. I will talk a little bit centenarians myself in my presentation, but I think maybe we don't have this good this good measure to see, and of course your population here is really particular and healthy, besides many diagnoses, as you say, like quality of life, and they are coping. So besides diagnosis and like diabetes and using metformin, they're coping well. So probably we are not seeing this in human cohorts because it's too difficult. We have like several variables to measure. We can't.
Yeah, I agree with you. And when I decided to take a look in sarcopenia, I was scared because we did not have all the measures with the conventional techniques. But one thing that can be helpful in this case, all the patients were analyzed with the same cutoffs. And we did not see any difference comparing the groups. So even if there is something wrong here, it was not significant. And also, we don't have obese patients. We have some obese patients coming from the other outpatient clinics. But after 80 years old and independent, they are healthy. Yeah.
Yeah, so that's why I think it's really frustrating when we don't see the results. But I had this myself. I was conducting this cross-sectional study, and I had 4% of sarcopenia. And I was like, it's impossible. Like free living older adults coming from public services too, and it's impossible to have only 4%. But I believe there's something wrong with the the measures that we are using. So, but I agree with you. Your population is really different, and I hope you can find something now that you are receiving people from other services. But I believe it's the epidemiological scenario we have for older, like 80 plus, and also the cutoff problems that we have in.
But one good thing in favor to cough circumference is if you detect that it's not a good measure and you increment diet with protein and start doing some resistant exercise, the patients improve gait speed, improve everything. So we can continue to measure and see if it increases, and it happens. So for our day-by-day activity, it's good, it's easy, it's cheap, And I think that we can trust this measure. So for follow-up, it's good. It's good and it works. So
Okay, thank you very
thank
much.
you.
So I would like to ask you, Gisele, thank you for this nice presentation. And well, my question is, first of all, I think you just said it's not real life. You have a very specific group of patients that are coming often to the hospital and they are concerned about health. So my question is, do you have the record of their style of life? Like they do any sort of physical activity, or what's the diet of them? They do this intermittent caloric restriction or not? I mean, just few data that maybe you can have a guess on how they are doing so well, more or less.
Yeah, we have this data, but it's not consistent for everybody, so it's not possible to analyze because we missed a lot of information. But caloric restriction is something that old people do. They eat small amounts. So sometimes you have to increase the amount of protein, amount of calorie, because they don't eat enough. After pandemics, there is another problem because they don't afford food. And sometimes they cannot eat the sufficient amount of protein because they can't. So the only thing that we can do is improve the quantity of calories and not necessary protein. So in this terrible scenario, we are expecting that they are performing badly in terms of gait speed, in terms of calf circumference, and all the measures for sarcopenia, but it's not happening. They stay like stable, okay? And so caloric restriction is a fact because they don't eat that much. But fasting in the way that we know currently, no, they don't do. Sometimes they prefer not to have dinner. They like to have milk and a slice of bread or soup very early, and they don't eat much at night, and then they sleep. And after they wake up, they eat a little bit more in the breakfast and at lunchtime, and maybe some snack in the afternoon. So they do some kind of they have some kind of good habits. It's not intentional, but we see that they don't eat much. They are skinny, they are not fat, and they don't eat that much at dinner. And we have to insist sometimes to have some supplement, protein supplement at night, at least to have protein if they can afford, but most of them can't.
Well, I was just going to ask you about the vitamin D. Have you done any?
Yeah, all the vitamins, we are looking at them like crazy, like vitamin D, B12, and folic acid and ferritin. So we pursue all those elements to be sure that they are okay, because all of them are important for everything and also for cognition. So they are very compensate. Most of them have osteoporosis, so they receive treatment for osteoporosis and also vitamin D in with a good quantity, because they do not expose to sunlight so frequently. So vitamin D almost 100% take.
And I think it would be very nice to study the immunological state of them, because as Daniela told, when you are approaching centenarian, you get like stable. If you don't get any cancer or very terrible disease, you get like stable and you go. So maybe on the immunological side, we can find the same. They are not different from each other. I mean, they will have a immune profile that can help them
Protect
to
them,
protect
yeah.
them from infections or cancer.
Previous studies from Dr. Maiza show that they don't have an inflammaging in the same way that we see in obese and diabetic patients at 60 years old, for example. Yeah, they don't have. So this is not a problem. They have all of those disease related to inflammaging. But if you measure the C-reactivity protein, for example, it's normal. And also, they perform well after vaccination. We didn't have too many cases of COVID and influenza because they are vaccinated. Every consultation, the vaccine board is checked, so we know that they are taking. If not, oh, you have to go. So
Yeah, very controlled.
yeah.
Yeah. Okay. So I was just wondering about Alex patients. Are they old or what's the age of your rheumatoid arthritis patients?
This is an intriguing question because we see all ages. Usually they are middle-aged when they get the disease, but with the improvement in therapy, they are surviving longer. So we have old patients with rheumatoid arthritis with lots of comorbidities because of the burden of the disease or even chronic use of glucocorticoids, but they do well. I mean, they survive much longer than in the past, and cardiovascular disease is the concern about the survival. Rarely they develop extra articular manifestations, something like rheumatoid vasculitis or pulmonary nodules, or even rheumatoid nodules. So the disease is better controlled nowadays with better therapies.
We have some patients with rheumatoid arthritis, but all of them are controlled without any therapy. So they had some time in life, but currently they are without activity. So this is a very different population. They are very well controlled. This is the message. After 80 years of age, from 80 years old forward, probably looking at all the aspects together can make the patient survive better with less uncompensated comorbidities. The comorbidities are there, they have, but they live well, taking a lot of medications. The polypharmacy is something. And when you see all the historic of diseases and polypharmacy, you see how can they live taking all of those medicines, but they are doing well until dementia. Dementia is the worst thing that can happen.
That's a problem. Yeah. We can see nowadays relatively young people with developing comorbidities with Western diet, becoming obese and developing hypertension in their 30s or 40s. And we wonder, for example, for sarcopenia, in which you have an inflamed patient with lots of IL-6 and low bone mass and also muscle mass. We can see at what age it would be feasible to start, for example, exercise at a high protein intake in their diet to prevent this. Should it be at the lifelong work to prevent, or during the 50s or 60s to prevent the development of sarcopenia?
Yeah.
What do you think about that?
It's a very important question. You know, I'm just working for 2 years in this scenario and reading about this. But I think that 40 years old is a mark, because all the metabolic syndrome will start the development in this age for men. And if you can keep a good muscle mass without obesity. You will not have hypertension, or if you have, it will be easier compensated. And all the cardiovascular alteration, lipid alterations, they will be fine. You can take your statin, okay, but if you start in this age to take care of yourself, it will be fabulous for the rest of your life. And for women, 40 years old is the beginning of the premenopause. We know that it starts around 40 years old, even with very slight symptoms. And one of the reasons that I make this career shift is I was sick to see my hematology-oncology patients suffering from metabolic syndrome. I know that inflammation is the cause of cancer. If there is another known cause, I know that this is the reason. So after treating lymphoma, you have all of the other disease to compensate, and it's very hard. And the other cause of making this shift, it's my personal journey after menopause. I changed all my lifestyle, and I know how it changed all the aspects of my life. And so I believe that if you don't take care of you until 40, you have to do this. It's an obligation to take care of you after 40 years, independent of all the other disease that you can have, even with rheumatoid arthritis. Maybe at 60, you will not have the disease in such activity. So we have to remove the inflammation from our body. This is the most important message for longevity. And you can do this in many ways. And caloric restriction is a way, and fasting is a way. That's why this is so popular. And doing exercise and resistance exercise, it's very important, because it's important for your body, your health, for to avoid inflammation, and to your brain, because Alzheimer does not start at 60, it starts at 40. So you have to prevent this. So the best time is when you are 40, you have to start take care of you. And there are many coaches, many people doing this kind of work currently, but a geriatric doctor can do it easily. Maísa, do you want to say something about this? Even in Portuguese. I can translate. No,
Say something.
O que você acha sobre começar a se cuidar o mais cedo possível?
Eu acho que a geriatria é o estudo do envelhecimento. Então, na verdade, a gente não começa a atender o paciente quando ele é idoso. A gente começa a pensar no envelhecimento quando a gente nasce. No certo era a gente nascer, sair do pediatra e ir para o geriatra. E o geriatra ele faz a gestão da saúde junto com outros clínicos, outros médicos, mas o geriatra está ali como um ponto de apoio para o paciente e para a família. E essa parte da inflamação, a gente vê que todas as doenças que são incapacitantes no idoso, muitas vezes está relacionado ao inflammaging, que é muito importante a gente conseguir barrar quanto antes a gente entrar num estilo de vida mais saudável, com exercício resistido, com tudo que a professora Gisele falou, melhor vai ser nosso desfecho e nossa qualidade de vida, com 80 anos ou mais, né? É isso.
I'm too late for that.
No,
It's never
I think
late.
anytime is time to start. If you start, you can get something. If
It's
you
never
don't,
late. Even for 80 years old patients, we can do something. So we have to do something for ourselves. And we are always working and taking care of kids and taking care of family. And sleep, it's very important. We have to learn how to sleep. Using or not melatonin, for example, I have to use melatonin, but no problem. Now I can sleep and I feel better. So we have to find ways, and there are different ways. And for you, it's something that is very personal. It had to be adjusted for all of your
just put a quotation of these releasing kids because they are not intended to be teaching, you know. All the reactions you can do is really different to because it's a factory. How many of the major products are but not but the reactions are their enzymes activity measured. So if you have the different, for example, either you can measure it using tryptophan and using the rate, so you can measure those two, and then also the enzyme activity. So it is very interesting. And see the samples. Not that, but yeah. You can do it in PCR. It's not very expensive in the kind of something. So you can do it for 6 months. Which is fine. Maybe you already know. Yeah, I already know. Yeah. And probably consequences so big this for everybody, it's the process. It's not separate. Taking a look on the biomarkers for health. So we have short-chain fatty acids. Yeah, that's I saw in one of the articles. Yeah, that's another. We also found. Yeah, I found like 5 or 6 that would be interesting to measure. So I think that now we have data to pursue all of those markers. And 60 and scales.
So now I'm very pleased to have here Professor Ligiana. She's from Unicamp. And thank you very much, Ligiana, for accepting the invitation and to come here from that distance. Thank you.
Good morning. So my name is Ligiana. I'm professor at Unicamp. My area is nutrition, and I'm 40 and something years old, and I have 2 small kids, so I don't sleep. I don't have time to practice exercise. I'm always stressed, so that's why I was like, it's better to get out of this section, you know. But just kidding. Well, I'm talking about diet and healthy aging. When I was preparing this talk, I was trying to figure out how can I contribute here, because of course you all have great researches about aging, and but I think we should discuss diet in some points from a nutritional view, right? So I hope it can add something to this section. Our syllabus today is why diet is always on focus, first of all, and then I'm talking a little bit about longevity and diet. I'm bringing some evidence from science to Netflix. I'm just talking about supplements, if we can find shortcuts, and something about future challenges and take-home messages. Well, first, why diet is always on focus? Food is always involved in research on longevity. We have frequently choosing bad guys and good guys, and this research area is always changing. So first of all, you can eat a lot of eggs, and then you cannot eat eggs. Then you can eat now. So it's like a changing area that encourages several misunderstanding and taboos. So food is always involved in research at this point. I brought here some several examples of books. It's not scientific books, of course, but several books that promotes foods that can make you younger, that can bring you longevity, but in the core, we are not looking at the right direction when you just talk about 100 foods for keeping you younger, or Chinese foods for longevity. We have to look more broadly when we talk about nutrition, right? So I have here this example that I love, that it's a woman that was turning 110, and she said that her secrets for longevity is drinking 3 bottles of wine, of beer, and 1 glass of whiskey every day. So I'm going for this one, because it seems better than healthy diet and exercising. So let's stay with Miss Agnes, that it's probably easier and funnier to have like longevity like that, right? But actually, when we look at the Chinese secret, it says like the longevity secret is eating a half, walking the double, and laughing a triple. So this is probably, it's a really antique saying, that it's probably one of the things that we are discussing here now. I myself prefer Mrs. Agnes' approach here, but probably we have to stick with the Chinese saying. Okay. I like this graph. It's from a publication a little bit older, but I like this graph because it brings 12 preventable causes of death in United States that can be controlled with food. Okay, so we see like high blood pressure, overweight, high glucose, LDL cholesterol, high dietary sodium, salt, and salt appears here again, low omega-3, high dietary trans fatty acids, or modified because we don't have trans anymore, low intake of fruits and vegetables, and low polyunsaturated fats here, that it's going that direction of omega-3. I also call the attention for the alcohol use, that it seems to be associated with some preventable deaths when we talk mainly about injury or other diseases. But when we talk about cardiovascular disease, at this point in the beginnings of 2000, we had this body of evidence saying that it was probably avoiding cardiovascular diseases than contributing to higher risks, right? So we are talking about mainly diet and lifestyle here, right? But diet can play an important role in health and longevity, since several nutrients and dietary patterns can be anti-inflammatory, and we are talking here about inflammation the whole morning. So probably diet in some scenarios is acting preventing or controlling inflammation. Well, I find this paper really interesting, that it's from 2020, that says like nutrition and longevity, from mechanisms to uncertainties. And then they put some things that they consider, it's a review, it's a broad review, and they consider some aspects that already established, like whole grain intake and vegetable and fruit, nuts, lower intake of red meat and mainly processed meat, healthy diet intake, whatever this means to us, And they caloric intake lower, that we are already discussed a little, and 2 points that they say that we need more evidence. But when we talk about methionine, we are usually talking about red meat and processed intake. So I believe there's not much to discuss here, but there is this point that's pyrimidine. And I will focus now this first part of the presentation about caloric intake and red meat and processed meat. Well, we have this really interesting paper here that Professor Daniela is also in the authors here. They say it's aging and chronic inflammation. Highlights from a multidisciplinary workshop that it brings some highlights from a workshop that was in 2022. They point to mainly things here that I would like to highlight. First, consumption of polyphenols, mainly from cocoa, our cacao, right? Flavonoids in fruits, vegetables, and cocoa can modulate important networks in genes and blood cells involved in functional process and interactions, probably with the vascular endothelium. And then they discuss a little bit about dietary restriction without malnutrition. It's important to say that may be the gold standard for delaying aging and extending life and health. In mice, most of our research, but it's important that these findings will be clinically translated into a personalized food as medicine approach. So when we talk caloric restriction, the first evidences in humans appear to be in World War II, that they noticed 30% less mortality compared to pre-war period when we see caloric restriction during war. And they also started to notice evidence from Okinawa that begins in the 70s, that they have around 17% fewer calories compared to the national Japanese average, and they have little weight gain during age, low BMI, and lower risks of mortality from diseases. So the critical points here in all those evidence that we have in humans is that fasting and high caloric restriction is difficult to achieve and maintain in general population. We have some evidence that bone mass may decrease in locations that are vulnerable to fracture, like hip, femoral neck, and lumbar spine. And the ideal caloric restriction value to achieve these anti-aging results, it's not easy to define, especially in older adults, because we have the challenge that how we prevent muscle loss or sarcopenia in aging if we are restricting calories and proteins, of course. I'm talking about proteins also. Well, I brought here some discussion that comes from another review paper that says that we are talking here in humans of a mild caloric restriction that says adopting 11 to 12 hours time restricted eating, as we are talking about earlier, like you stop eating from 6 at night and start eating again in 6 in the morning. So you intentionally put time restricted eating of 12 hours. And it's not that difficult for our culture in aging, right? They usually do that. But we have as well few yearly cycles of 5-day fasting or fasting mimicking diets, they call FMDs, that may be alternate day fasting or fasting for 2 days a week, that we call intermittent fasting. This is not that difficult to apply in humans, but we don't have enough evidence to say that, oh, yes, this is the way we can achieve aging and also not not promoting sarcopenia or not promoting loss of muscles, and because this is really tricky when we talk about epidemiological studies, right? So this is another review from Cell that it's 2023, I believe. I'm not seeing very well here. I'm sorry, it is too small. But they bring this really interesting figure about the longevity diet and the aging characteristics that can be related here. So what do they point? That first, the longevity diet should contain mid to high carbohydrates containing, let's say, 45 to 60% of our diet. Low but sufficient protein intake, we say 10 to 15% of our diet, but they call the attention that it's mainly plant-based. With a regular consumption of pesco-vegetarian derived products. What it means, low in meat and processed meat, and mainly plant-based proteins. And also 25 to 35% of fats, also mainly plant-based. And I am talking about Okinawa again because I'm bringing, after that, the Netflix evidence. So animal products represent around 1% of the traditional diet of Okinawans. And occasionally, they intake meat or animal products also that characterize populations of Sardinian locations and Loma Linda in United States, that they are known for centenarians or high average lifespan. So when we say about protein restriction here, I'm not talking about let's eat less protein, but let's be more wise choosing proteins, right? The pros about protein restrictions as that first, and several evidence we have that in rodents have shown life-prolonging effects regardless caloric intake. So you reduce protein, you can prolong lifespan. This was observed in a really big study, epidemiological study, that's called Envelhecimento in the United States. I believe you all are familiar with. And they noticed that adults from 50 to 65 years old with higher protein intake, around 23.7, 24% of the diet, had 74 increase in overall mortality and more than four-fold or 4 times risk of death from cancer when compared to low group protein that was around 8.5%. When I saw the first time this data, I was like, what? Because for nutritionists, we cannot think about a diet with 8.5%. It's too low for older adults, right? For if we follow our recommendations nowadays. But these effects are determined by animal and not vegetable proteins. So if you have a higher than 8.5, for example, percent of consumption, but with vegetable proteins, we did not notice this increase in mortality. Okay, so the cons, the low recommendation, like I said, for protein restriction at first, the low protein and carbohydrate ratio also increased body fat and resulted in hepatic steatosis in some researches. And in the same study, in Haynes 3, they noticed that from 65 older, the high amounts of protein, around 28% of the diet, can be assessed, seen as a reduction in all-cause mortality. And so this seems tricky because I just said that we have to keep it low, but now I say that no, we have to keep it really high. But they noticed that because several guidelines we have in nutrition and in older adults, for example, Aspen guidelines and the Protage group, we say that we have to have at least 1 to 1.5 grams of protein per kilo of body weight per day to maintain muscle. So we have a kind of problem here, right? But if we come back to these first results, let us remember that this is not determined when vegetable proteins are involved. So back in this graph here of this first paper that I showed, when we say healthy diet here, we are talking about whole grain intake. We are talking about vegetable and fruit intake. We are talking about nut intakes. So I'm going to call you now outside from the box of proteins and calories, and I'm going to start talking about diet patterns or dietary patterns, right? What is a healthy diet intake? This is really subjective, but I'd like to bring some of the recommendations from this document here of WHO, World Health Organization, that says risk reduction of cognitive decline in dementia, and then bring 3 main recommendations around the diet. They first say the Mediterranean-like diet or similar to Mediterranean-like diet style may be recommended to adults with normal cognition and mild cognition impairment to reduce the risk of cognitive decline and or dementia. The second recommendation would be healthy, balanced diet should be recommended to all adults based on the WHO recommendations or whatever recommendation we are following here. 3, vitamin B, E, and PUFA and other multi-complex supplementation should not be recommended to reduce risk of cognitive decline and or dementia. And what they say, it's like the main point of this document in the nutrition section. They say a consistent healthy eating pattern throughout lifetime, or at least during aging, let's say after 40s, it's more important than the role of a specific food or nutrient, especially if offered as supplements or medical foods. So what I'm talking about here, when I say dietary patterns, we say we have an operational definition for what is a dietary pattern. We usually say the set of group foods consumed by a given population, but we are talking about 3 big aspects here. First, eating occasion patterns, how we eat, when we eat, what is the frequency of our food intake. Second, eating format or content, what nutrients or foods are involved in our regular diet. And then the context, who is with us, how can we have this food encounters with others, with friends, with family. So these 3 key points talk about our dietary patterns. So it's not like, okay, I know that Mediterranean lifestyle is good, so I'm trying to eat as much as olive oil I can afford, because nowadays it's really expensive, at least here in Brazil, I say. And now I'm like Mediterranean. No, it's not like that, because we have these 3 big concepts here involved in dietary patterns. So when we say about the Mediterranean, that it's the most known, the most studied for increasing longevity, we talk about intake of vegetable and fruits, but that brings us like good fats, omega-3, that nuts, olive oil, and fish. what we got from here from in Brazil and several other Occidental countries, we decided that we had to eat a lot of fish, but we forgot the nuts, we forgot the olive oils, and the whole pattern, right? Low intake of meat and its products, and also dairy, low glycemic index carbs, and also high fiber intake, and moderate consumption of alcohol, mainly wine in this case, because first they bring phytochemicals, vitamins, minerals, phytoestrogens, and anti-inflammatory agents, and mainly in this case resveratrol, that is found in wine mainly, and this is one of the most studied polyphenols with antioxidant properties. So let's go to the other side of the class, that we're going to Netflix. That this is really talk. I believe I heard like 5 or 6 talks about the blue zones in the last year since the Netflix documentary was launched. I don't know if everybody here is aware or watch it, but they call the blue zones a region that were higher than usual number of people that live much longer than average, and they point 5 blue zones. First, they talk about Okinawa, that is probably the most studied, the longest studied, and then Icaria in Greece, Sardinia in Italy, Nicoya in Costa Rica, and Loma Linda in United States. I'm going to pass really quickly here of the main characters of these blue zone diets. First, if we see Okinawa, they have a lot of carbs here, come from sweet potatoes and rice too, but much less. Fish, meat, and poultry like 2%, 1%. Other foods also 1%. They have a lot of vegetables, other vegetables, and legumes. We say in Brazil, grains. It's not like the green vegetables, but it's higher in these grains. In Icaria, Greece, we see a lot of greens, fruits, legumes, even higher than in Okinawa. Olive oil, fish, they have fish here. It's much higher than in Okinawa, for example. Pasta to meat, 5%. It's a little bit higher too. And sweets, oh my God, they have sweets, thanks God, right? But they have so many well-distributed vegetables here that it's even difficult to point. And then we go to Sardinia, so probably the same, right? No, they have a bunch of pasta, but it's mainly come from whole grains. Pasta or bread, they are usually made with whole grains. They have a lot of dairy, so it's not the dairy the problem here. They have vegetables, but they also have a small amount of fish, legumes, added sugar, fats, and fruits. You see that it's not that rich in fruits if you compare to, for example, here, 16% in Greece. So I'm not following. If I'm just looking at these graphs, I said I'm not following. So what is the food that I have to put in on my plate? It's pasta, it's fruits, it's sweet potatoes. So let's stick with the sweet potatoes and chicken diet. It seems like, but the other places say no, it's not like that. Well, let's go so to Nicoya, that we see like whole grains, dairy also in high amounts, vegetables. We have a lot of sugars, like not a lot, but 11% is higher than the other locations. Fruits, legumes, and we also see small amounts of meat and eggs here. And in Loma Linda, we see a lot of vegetables and fruits, much more than the other locations, and legumes, mainly soy. We see also dairy in whole grains, most coming from oats and not wheat, as Italy. Added fats, added sugar, really low, and also fish and eggs. So how can we translate that into our our plates, right? We can say that, long story short, we have plant-based diet, not 100%, but almost 100%, let's say 95 to 100% plant-based, low meat, and limited added sugar, salt, processed food. And what they have in like variability, they have the distribution of food groups or macronutrients. So this is not the point here. It's not like I have to have 15% of proteins or 50% of carbs. It's not the distribution that makes sense here, but what we have in commonalities around low meat and low added sugar and salt and fat. If we see the graph that is presented during the documentary, they say that the Okinawa recite a phrase before every meal that says "hara hashibu." It means stop eating when you're 80% full. So you don't stop eating when you say, "Oh my God, I'm full." No, you stop eating before that. So they call the 80% rule. Maybe this is a good advice for us. They also say wine at 5, that it's enjoy a glass of wine with good friends each day. You see that they say at 5, not at 8, not at 11 o'clock, like I'm working hard until midnight and then I decided to fool myself of wine. No, they say it's more than wine, right? It's stop working in a reasonable hour so you can have a chat with your friends and enjoying a glass of wine. It's much more than lifestyle than just the resveratrol as a personalized good guy, you say. And also plant-based and vegetarian data said that it's around 95% plant-based, all these diets. They also say that it's just that, no, we have 4 areas in this graphic. We have move naturally, and I added or artificially, but just do it, right? They point like work by hand, gardening or other light activity, and walk. All those 5 locations that they studied, they were upper hills, so these people walk a lot. If we talk about connect, it's also very important. Put family first, partnership, and find your right tribe. Let's say I like to be plant-based. I decided to adopt really plant-based diet, but my husband likes to have a barbecue with friends every Sunday. It's not probably my right tribe. I will have to struggle to keep it. So they say you have to put yourself in a group that has the same values and the same lifestyle than you. And also outlook, like unwind, faith, and purpose. If you don't have a purpose in life, if you don't have anything to come back after work, family or your hobby or something else, it's not probably a good diet. So as I was talking about first, when we talk about healthy diets or dietary patterns, we talk about a lot more than just a diet. Do we have a blue zone here in Brazil? Yes, we have. I don't know if you are aware of, but we have a place called Veranópolis that in Serra Gaúcha. It's being studied like for like I think 20 or 30 years. I believe 30 years now. They are studying, and they are known as the land of longevity. They have 24,000 people living there. And they also received some representatives of the Blue Zone Initiative to understand if they could be in the next documentary or the next book about the Blue Zones, right? What do they have in common with the other Blue Zones? The cities over the hills, so they are the physical activity is high. The food is mainly natural, very little processed, and they have many social encounters, especially linked to spirituality, that links to purpose and faith and whatever I told you before. So if somebody say for me, for example, today, you have a disease, and I say, oh no, well, it can be cured with diet and exercise. And I will be, oh my God, oh no, I will die anyway, because it's too difficult, right? To take this healthy lifestyle for us, living in a huge city, like São Paulo. I left the huge city, but I also have living in cities and low time to physical activity, low time to eating well. So how can we do that? Can we have a shortcut? Well, let's try some supplements, right? Sorry, no. We have a lot of evidence saying that supplements alone don't bring light to our darkness here. But I brought these two examples that I believe it's really important. This is a review about the influence of omega-3 and dementia. And they also have those responses meta-analysis. So it's really beautiful study. The main results here is that they found the beneficial effects tends to increase in the first 12 months of intervention. If you take daily more than 500 milligrams of omega-3 here. But oops, I'm sorry, this is not in English. I forgot to translate this part. The effect was not general. So in the countries where the blood level of these compounds are really low, like Brazil, for example, these beneficial effects are not significant for cognition and function, executive function. So it's not like we Brazilians don't eat at all omega-3, and then we decided to take a lot of pills, and the effect is not the same, right? Probably because the activation of several genes by this dietary omega-3 may also play a crucial role in facilitating phenomena occurring in perinatal period and a gradual progression. So it's not like we are taking these pills after 40 and we are going to prevent dementia only taking this omega-3 being zero exposed during life. The more the better. No. The dose response curve was downward sloping after this 12 months of intervention in most of studies. And when a dosage exceeds 420 milligrams of EPA. And also they noticed that people with normal cognition, the effects were not actually that good. Mainly if you don't take these daily doses, it's not different from daily and total doses of omega-3 and EPA. The side effects that some previous studies already suggested that a high intake of omega-3 may be actually associated with LDL cholesterol raising effect or a decline in platelet here. I'm not, of course, entering this discussion because I believe you here are much more experienced in this area. And the mechanisms, they say that probably influencing synaptic plasticity and neurogenesis in brain regions that are susceptible for oxidative stress. I also have this study here that I found that's really interested in. This was published in this year, the impact of dietary supplements in blood pressure in older adults. And they analyzed a whole bunch of kind of supplements here, like vitamin D in 2 dosages, resveratrol in 2 dosages too, and other. They noticed that none of them were different from placebo in general, reducing overall blood pressure. And they conclude that in general, the methodological rigor of the studies are really low, and more studies are needed to determine if we need a different doses and combination of dietary supplements to get any interested effect in blood pressure. Well, for future challenges, I'm here pointing something for research, for professionals, and for individuals. I believe for research and professionals, we have to still understand the in vitro and animal results and translate them into safe recommendations for humans. I think this is the most difficult part of the science. We have to clarify the role of nutritional supplements as a possible substitute for long-term diet style or consumption. It seems like we cannot do that, but we still have many studies ongoing, so maybe we have some more evidence in near future. And we have to translate all this research in public health policies to help other populations to be blue zones. We don't have to have only 5 or 6 blue zones in the world. We have to have scientific evidence walking together with social structure and political will to construct decent public health policy here. For individuals, I would say adopt the motto unpack less and peel more, that is preferring natural over processed food. Change the lifestyle in general along with the diet. So as you said, sleeping well and exercising and other changes in general lifestyle, so we can benefit from better diets. And also we respect and try to follow updated guidelines, such as our dietary guideline, Brazilian population and it's published almost 10 years ago now, and is one of the first food-based dietary guideline and not based in nutrients. So Brazilian guideline is talking for 10 years now. We have to prefer natural, we have to have company, or we have to have our right tribe to make more pleasant eating, and we have to take care of our food more like a healthier lifestyle than one nutrient specific. So my take-home messages here, it's not mine, it's from Veranópolis. This is a paper piece that it's from 2016. I don't believe it's really clear, but it's almost 10 years ago now too, when Veranópolis was really discussed in open media. And they say, I have two things here that I would like to take home messages. First, a couple of 90 and 80 husband and wife, they were together almost 60 years back that time, and they said they avoid the trash food of the supermarket. They have cattle, chicken, and pork produced in their property, and they plant legumes and vegetables. So they eat natural. All of them, almost all their diets, it's from the garden, right? And they also produce and consume their own wine. 1 glass in the supper and 1 glass during the lunch. Their own wine. It's not like they buy wine, right? So I believe this is really beautiful to see that we have these people going to 80 and 90s and still planting their food and making their own wine and sticking together as a family, as a community. And also the other one that says here that the secret for longevity is taking a life without exaggerating, don't stick still or movement, and mainly love much. Without love, it explodes everything. So they are talking here about not about a diet, not about only the movement or physical activity, but they are talking about belonging, family, and community, and all these things that we have to look at when we say we are going to have a healthy diet. Take-home messages here. It's for research. We still have to figure out how to promote this caloric restriction and protein restriction in older adults. Supplements can be a good alternative for older persons that cannot eat properly, but no, they are not shortcuts. Taking one pill and we have it all solved. Dietary pattern seems to be our secret of longevity here, mainly the plant-based and lifestyle. Centenaries usually have, besides healthy diet, they have stress control, purpose, faith, regular simple physical activities such as gardening or walking, and social support. And this couple was also from Veranópolis. They were turning, I don't know how to say that in English, but they were together, married for 70 years old in the day of this interview. They had 7 children, 15 grandchildren, and 7, how is that in English? Grandchildren. Yeah, I say that. So they say he was blind. She was partly deaf, and they say he was cutting the wood, and they were together constructing vine things, vines, objects, and they were still selling these objects in the city. So probably they found here the secrets for longevity and for also a happy life. Thank you very much. If you have questions, I also have many.
Thank you, Ligiana, for this very nice presentation. So you brought some secrets. They're not secrets, we know already, but how can we cope with them in our busy life? You said you left São Paulo to a small city, but still you have all this rushing all the time. So anyone would like to make a comment, a question?
Yeah, very, very interesting. And actually, this is what I'm teaching in my class of centenarians. So the 5 areas in the world where I also include Sicily together with Sardinia, because there are 2 distinct groups of centenarians. And what is important, and I'm also saying, is not only the healthy nutrition, but is the healthy lifestyle. So this movement up and down the hills, and the social life. Social life means faith, whatever you have said. Yeah. And this also is very connected to a healthy immune system. So I'm doing it from the other side, but I totally agree. I have a comment on the omega-3 study, in which they did this extensive amount of milligrams per day for a long period of time. I think that equilibrium is the most important thing in life. So it's like the intermittent fasting, the fasting mimicking diet. All these things have the senolytic treatment. You have to take them and then interrupt and give time to your system, your body, to get used to that intervention. If you do 1 year of omega-3 and rich food, this is what you get. So yeah. Not, the results that you were expecting. So it's not only the amount and the prolongation, everything is the equilibrium. It's like the keto diet that now was very famous until last year. And everybody was using this keto diet. My God, is dangerous, because keto diet is 95% of fat. And you cannot take it forever. You can take it for a few days per month. Walter Longo, you cited his review, is actually fighting against the keto diet because of this, because you get more fat that you can process. So your body has the organs and tissues floating in lipids, which is extremely deleterious. So I think that the take-home message also is everything with parsimony and equilibrium in life.
Yeah, everything in moderation, right?
Well, Ligiana, I would like to ask you a question because I know you were working at SABI study before, and it's a huge population of old people. It's not controlled as Gisele's and Maisa's. It's like everybody or representative population of old people in São Paulo. So in Limeira, you have the same sort of study, or what you're doing since you got there? Because I'm curious.
No, in Limeira we don't have that. Since I left my PhD and I finished my PhD and went to Limeira, I started to look at dietary patterns. I conducted a study that was about 600 older adults from Limeira, Piracicaba, and Campinas, but it was cross-sectional study, and then I got pregnant twice. Mainly in the middle of the pandemic period, so I did not have time to do anything else. And now I'm working with Esse results, that is longitudinal study of aging in Brazil, but we don't have much data about diet in these big studies, SABI or Esse, because the questionnaire is so long that there's no time to ask about diet. So in my small cross-sectional study, I started to look at this dietary patterns, and what I noticed is a little less what we already discussed here, like this fasting around 11, 12 hours, that is really common. They stop eating around 6, 7 in the evening, and then only take breakfast. So it's around 11 and 12 fasting period. But we say the quality, the overall quality of the diet is really low. They are already exposed to these ultra-processed foods that before we did not notice, but it's much less than in younger populations. But they also have low socioeconomic status, so they are not eating that much of meat. But my main concern in all these observations is that they are also leaving our rice and beans combination. That is one of the things that we have in our Brazilian food guide, that it mimics all of those plant-based protein diets, because when we talk about rice and beans, we have like several combinations of amino acids that are really good for health. So I noticed that in these other populations, like in Limeira, Piracicaba, and Campinas, we still have the rice and beans culture, but it's going less and less frequent. And this is really a big concern for, I think, the epidemiological studies in diet. Also, all of them are really concerned about this, lowering this our Brazilian pattern, because this is a good protection pattern. So that's it.
I have the microphone. Well, great talk. I have 120 questions, but I will only ask a few. It's interesting that the literature is showing that it's better to choose the food instead of supplement. So instead of nutrients, instead of taking omega-3 pills, eat fish. But coming back to the first TV show that you referred to us, the twin experiment, you are what you eat, the twin experiment at Netflix. When I watched it, I had the impression that they want to preach that if you have a vegan diet, it would be even better than the Mediterranean diet or something like that, because they said bad things about farmed fish. At least I try to eat fish almost every day. I love it. But for example, if you go on a vegan diet, then essential amino acids, how can you get them? You need to have a diversity of different types of food to get those amino acids. So it would be much more difficult. So is there any comparison between the vegan diet and Mediterranean diet regarding that?
I would say that most of this blue zones region that we saw, they are almost vegan, but they are not vegan, right? Because they use it depends on the place. They have dairy or fish or whatever, but they are 95% plant-based. And when we talk about proteins, we have to be aware that our main source of proteins is grains, beans, right? So all these diets, they are really rich in beans, around 1 to 2 cups of whatever beans you like each day. But it's probably best if you can alternate those beans so you can get a lot of different amino acids. And in Brazil, we have the rice and beans, really good combination. We have almost everything we need from rice and beans. The point is that we are not eating anymore. But yes, there's a lot of research saying that it is possible to go plant-based. You don't have to be vegan. You have to take it low with meat or even fish. We believe fish is really good for health, but here in Brazil, for example, we don't eat real salmon, right? We eat a Chilean salmon that it's not free-living fish, so it's not full of omega-3. And then we get this pieces of information from media and says, now I'm going to eat fish because it's really good for health. It can be, but we don't have to put like, it's what I said. It's not one thing, one food in specific or one nutrient, but the whole diet pattern. And all the research is going to the place that like go almost vegan, but you don't have to be vegan,
Great. And the other question is related to the intake of whey protein that athletes love to do it.
Yeah.
Is it better to eat beef, chicken, fish instead of getting the whey protein? Or what do you think about that? Because it's something like a nutrient instead of a food that you're taking, and when you get that.
Yeah, take for example a beef. If you eat a small portion of red meat, you get several vitamins. You get iron, that it's really important. And I don't know you, but I like eating. I don't like only drinking. I love eating. So chewing, it's important. So yes, whey protein or whatever supplement we have, it's really important as a help even if we talk about aging. Sometimes you cannot get all the protein you need, or because you don't have the money, or because you cannot chew. So you have a lot of problems around that. So yes, you can use whey protein, but of course it's much better you eat the milk or milk derived products, low fat, dairy, or a beef, or whatever you choose of animal protein, then drink the whey protein. All the supplements should be helpers and not like the main thing of our nutrition based, you know. I don't know if I answered quite correctly, but I'm not really a huge fan of supplements. But as a clinical nutritionist, I usually adopt supplements when it is really important. For example, older person that it's sarcopenic, that cannot chew anymore, that it's really less appetite and whatever. So I use it, but I'm not taking that as like a flag.
Mm-hmm. Like an add-on therapy. Yeah?
Yes.
So if you have a base diet.
Yes.
Just a testimony. My boss, the full professor of rheumatology, she's 70 years old. She's very productive, but she burns the midnight oil every day. It's a slang for people who work until late at night from 7 a.m. to midnight every day. She's diabetic. She eats a lot of meat and carbs, but she's very healthy and productive at 70 years old. I think it's not an example, but she's out of the curve, of normality curve. Yeah.
Yes.
But I think in her case, the work renews her energy. It's not for everybody.
Yes. Yeah. Sure. Then we started to see individual cases and not like the epidemiological studies that's big groups or review papers or whatever, but we all have these excellent examples of not doing really good and still going really healthy, like that one that I showed. Like I wanted to drink 3 bottles of beer and a whiskey every day and be healthy, but I believe I cannot. My system doesn't work like that way.
You have another question? No. I didn't know that Brazil has this habit of rice and beans.
Yes.
And maybe before more than now. But I was last month in another immunosenescence meeting in Cuba. Cubans in Cuba eat only rice and beans. They don't have any money to afford anything else. Well, they have several studies that are connecting this kind of diet with longevity.
Yes.
And actually, they are very healthy. They are fighting cancers. Actually, there is no prevalence of cancer in the people that had for the entire life only rice and beans. So interesting. And these are data, of course, that are in Cuba. They are made by them, very good results, but will be difficult until they are published to see them around. So that's a very good.
Yeah, in Brazil we have this. We call it's what we learn in undergrad nutrition undergrad course, that it's like our main basic food for Brazilians is rice and beans.
Yeah. Well, legumes in general are so full in proteins and good proteins. But this study was done only in people eating, but only that, rice and beans. And actually, very, very interesting data.
Yeah, thank you.
Yeah, and it's a pity that less and less people are eating rice and beans.
Yes.
Let's hope we come back again. And I think we have to consider, talking about São Paulo or Brazil, that we are a big mix of genetic backgrounds. So you were talking about your boss. She's Japanese-based background. Yeah, so it's quite important.
From Okinawa, yeah.
From Okinawa. So we have, I think it's the second major Japanese population apart from Japan. So it's here in São Paulo, the place I brought you that day. And a lot of Italians, Spanish, everything. So it's very difficult to don't take genetics together with the diet, because, you can have a favor, some genetics that favors you to live more and without many diseases. But then if you go wrong, your genetics just abandon you.
Yeah.
But if you have a good genetic and you go right, even better. So you have
Yeah.
to consider, and we live in São Paulo in a very polluted situation. So many things to be considered.
Yeah. I don't know if you're aware of the studies of Bauru, the Japanese descendants in Bauru. No, we have just for context, we have a city around, I believe it's 300 kilometers from here that is called Bauru. It's a big city, but not huge, but it's a big city. And they have this big Japanese colonization. And one professor in University of São Paulo in nutrition, and of course then the group got bigger and bigger. They started to understand why these Japanese outside Japan, they had much higher diabetes than those in Japan. So they started to look at genetics along with changing the diet habits and also surroundings and whatever during generations. So they started studying those people that came during the war, and then the Sanseis, Niseis, I don't remember the orders of the generations, but they noticed that once you are here, you are already exposed to a more diabetic context. But if you can keep your diet as much as Japanese you can, you don't have this upper risks. But then the sons, the children and the grandchildren, as they occidentalized the diet, they had this high risk of diabetes, much higher than the parents and the Japanese in Japan. So it seems like, the genetic abandoned them. And they also said, okay, you're doing you wrong. Okay, I'm out of it. And it's even worse being here with a wrong diet than be, I don't know, Brazilian Italian or Brazilian, whatever comes your family, than Japanese. They just are doing really bad when they adapt to our lifestyle or our diet style. I believe the first studies, it's around the mid-90s, and they started to follow this cohort. It's probably in the third or fourth generation now. Yeah.
What's the name of the city?
Bauru. Yes.
It's just a comment about the Japanese patients in the Longevous project. I count 30 patients among 420, and they are first generation, and they came from Japan, most of them 80 plus. And one thing that it's very important is they are healthy. They don't have too many health problems. But one thing that calls my attention is they are not alone. And we have a lot of patients over 80 years old that live alone. So they don't eat well, they don't have anybody to talk. And dementia comes because of lack of stimulation. And those Japanese patients, they have family that take care of them. They live in a community. So probably diet is not the only fact. Yeah. Because it's a lifestyle.
It's like your
genetics is important, but it's a lifestyle. And the one thing that calls our attention is they are not alone. Mm-hmm. They always come with a son or a daughter that takes care of him or her, and they do everything in the right way. So this is part of the success. Mm-hmm. So when you ask me for healthy patients, I always think about them, but I cannot give you only Japanese patients. because it will not be real. And
Yeah.
There were some studies in the past showing that if you are, I think the Sub study or something, that if you are married, you can live longer and better than if you are not married anymore. Some of them die and you keep alone
Mhm.
the rest of your life because that's fact. You're not eating so well, you can get depressed. So, yeah,
Yes.
many factors.
Yeah. I have one friend that was in the PhD with me the same time, and she studied social support, not only couples, but social support in general. And her study was really beautiful because you see like a really clear survival curve that it's much better if you have a social support of any kind, not only couples, but if you have family around or friends or these church groups or whatever. So that's why all those studies is talking about you have to have a social support, and living alone, it's really a huge risk for health, nutrition, sarcopenia, and depression and dementia, everything. So thank you very much. I believe diet is not we never finish talking about diet, right? That's why I love this subject. But thank you very much for all the presentations and all the interactions we have here. It's really nice. Thank you.
Thank you for coming, especially if you're driving with problems on the car early in the morning. I'm sorry. Thank you for coming, even with the problems. And thank you for Ella. Thank you, Gisele. And thank you all of you for coming and asking questions, participate. And let's hope we can do it again next year. Yeah.
Professor, podemos tirar uma foto com todos ali na frente, fazendo favor?
A photo with everybody, yes.
Maisa is watching us, okay? Yeah, she's in view.
Isso, primeiro e depois todos, então. Aí, isso. E aí depois nós tiramos com a...
Yeah.
Ótimo. Agora todo mundo. Por favor.
So thank you, Prof.Ligiana. Thank you, Olivia, for putting this together. It was really nice.
Thank you for following us.
Thank you for coming back.
Yeah. So we should start doing something on like program of all these patients. You said you don't have many older ones.
I work mostly in vasculitis.
Oh yeah, you are in vasculitis. Yeah.
We are waiting for the medical approval to work on Pachias-arteritis and giant cell arteritis. So the same pattern, and large vessel vasculitis. One involves the elderly and the other young individuals. So to compare them.
Mine also are in the middle age. They're after the ones that they showed on Friday.
Yeah.
Those are middle age. Yeah. I don't have so many of them, just.
We have Professor Chaves. He's in charge of the rheumatoid arthritis outpatient clinic. We can talk to him to this collaboration.
Yeah, or maybe we can compare our